as I said, no calcium, but auxin, which is equally pleiotropic or even more maybe. And I will also like to talk about cell polarity a bit. And just not to confuse it with the last talk, this is Arabidopsis. Okay, so the system we were working for quite some time now, as I realized, is the movement of auxin between cells, so-called polar auxin transport. So auxin is unique among plant hormones, being in a regulated and directional manner transported through tissues. It might be true also for other hormones, but we don't have really convincing data for that. For auxin, however, it moves into the cell, it's somehow distributed in the cell and then moves out of the cell again, and then it moves in the next cell and so on. So that sounds like a very stupid way to move the substance through the tissue because it crosses basically all the possible membranes it can, which takes energy and which takes time. So if God was inventing this, he certainly didn't have a good day, one would say. But anyway, the advantage is that whatever signals each cell in the path of auxin can perceive and process and translate into the localization or activity of the transporters at the plasma membrane, that will then influence the flow of auxin to the neighboring cells. So each cell can simply say something about how much auxin will be flowing in which direction. And the cells can quarrel then with each other and compete, and then as a result of this coordinative action, then you will have a flow in a given direction. So the important feature of the flow is that it's directional, and the directionality was proposed to depend on the polar localization of the efflux carrier. The efflux is the rate-limiting step in the process. And then in 1998, there were 4 groups simultaneously identifying the family coding for these efflux carriers, so-called pin family. And the nice thing about the pins is that they are localized asymmetrically as predicted by the hypothesis. So if this is a cell of the longitudinal section of the stem, that would be the lower side of the cell, and that's only where you can find pin 1 in this case, and that's also in this tissue auxin is flowing down. So then we showed that the polarity of the pin localization indeed determines the directionality of the flow, and we showed that by several independent means. And then we started to use this system also to elucidate the mechanism underlying polar trafficking and cell polarity in plants. Well, the coordinated action of the transporters then results in many different developmental contexts in generation of asymmetric auxin distribution that you have here depicted by now already outdated DR5 auxin reporter. So that would be the shoot apical meristem, lateral root, embryo at the early stage and embryo at the later stage as example, and these colorful dots there, these would be the places of higher auxin activity. And if you mess up with the transport, you will mess up with that. And if you mess up with that, you will mess up with the developmental process. So that is basically the system. So on one hand, we can study the processes at the level of single cell, how different signals are integrated and translated into the localization of the carriers. We can also study the basic trafficking and polarity issues, and we can see it in the context of the developmental output, meaning the gradients and the developmental process. So that's the system, and I will guide you in one developmental example, how it works. That would be embryogenesis, and then I give you some more hardcore cell biology about the polarity maintenance in the plant cell. So embryogenesis, plants have embryos. That's my usual joke when I speak to the non-plant people, so saying always when you eat peanuts, think about what you are doing, right? And the embryogenesis in plants starts by the division of the zygote. So you have an apical cell and larger basal cell. Then you have, in Arabidopsis at least, quite uniform row of cell division and cell differentiation, which will then generate the basic body plant. And during the embryogenesis, you need to establish the apical basal axis, and you need to establish the bilateral symmetry with the embryonic leaves. So the two key moments for establishing the apical basal axis is just after the division of the zygote, this upper cell needs to get convinced to change the developmental program and starts to divide like that and not like that, how the zygote divided, and then generate the proembryo. So that's the moment when the apical end of the axis is specified. And then somewhere at this stage, the uppermost cell of the suspensor, which is the structure connecting the embryo with the mother, so the uppermost cell needs to get convinced again to start to behave differently than before. It divides asymmetrically, it generates the founder of the root meristem. And that's the moment when the basal end of the apical basal axis is fixed. And then the bilateral symmetry will happen, but I'm not going to speak about it. And that is, of course, the differentiation of the outer and inner at this stage, so the radial symmetry, which I'm also not going to speak about. So already quite a long time ago, actually, we could show that these two key events in the apical basal axis specification correlates with the high auxin activity. So at the early stage after zygote division, you will see higher auxin activity in the apical cell. So that's the moment when these cells will start to divide like that and generate proembryo. And then later in embryogenesis, you see high auxin activity again at the root pole, so at the moment when the root pole is being specified. And so at that time, we looked at the different pin proteins. So these are the transporters, these polarly localized pumps. And we found that if you look at the early stages, up to this stage, pin 1 here in red, which is specific in the proembryo, is not localized polarly and probably just equalize the auxin levels in the proembryo. And at the same stage, so meaning from the zygote, in zygote we don't know actually because it's quite challenging to do the visualization in the zygote, but at least from one cell stage, so roughly from that stage, the pin 7, another member of the family, is localized apically in the suspensor cell. So that would suggest an auxin flow in this direction, which fits with the accumulation of auxin then in the apical cell. So then, miraculously, something happened between these two stages. These are basically the same stage. This is 16-cell stage when the cell starts to divide already. And here we are probably just 10 minutes, 15 minutes later. So it's a very sharp transition. So in this moment, the pin 1 will polarize to the basal end of the provascular cell. So you see here is nothing, and here it gets to the basal end. And later, a bit later, the pin 7 will polarize to the lower side of the cell. So it will go from here to this end of the cell. And this then probably change the auxin fluxes, and this then generate the different distribution. So first we have the flow of auxin up by pin 7, equalizing by pin 1. This is the moment when the specification happened. Then at this stage, pin 1 polarize, reversion of the flow, and auxin accumulates here. And that's the moment when the root pole is being specified. And if you mutate the pins, instead of having then at the end of the day nice embryo with the apical structure and root, you will get just this ball which is dividing and not having any apical basal polarity. So that was really a long ago. And then we for a long time worked on other maybe more interesting or maybe less interesting things, but the question still stays. So how that seems from this sequence of events that basically the polarization of the pin 1 here is the thing which will trigger then all the changes leading to the specification of the root pole. So this will reverse the polarity of pin 7, that will reverse the polarity of the whole flow, and that will then give the signal from the proembryo to the suspensor to specify the root pole. And so we were looking a bit more closely to this, how this is regulated. If I say we, I mean Hélène, which is a great postdoc from France in the group, and she wanted to do embryogenesis, and so she started to work on that. We also collaborate very closely with Tolfeyers in Wageningen on this project. So the first thing what Helen did was actually, now she used the DR5 Venus nuclear localized, because you can see it slightly better, or she can see it slightly better. And then she could co-visualize basically then the activity of auxin and the polarity of the pins. And then she made quite elaborated statistic looking at the embryos at the different stages and counting how many embryos she sees which will have nuclear localization of the DR5 activity in the proembryo, and how many will have in the suspensor, and how many will have in the both sides. Maybe can you dim a light a bit? Would that be possible? Thank you very much, because even I can't see the DR5 activity in the proembryo here. Anyway, what she found out was that at the 16 cell stage, the change happens. So in blue, you have polarized pin, and in green, you have non-polarized pin, right? So at this stage, both things happen. So the pin starts to polarize, and at the same stage, you then start to see the activity in the lower, at the root pole. So it sounds like we have really a very tight correlation and sharp moment when these things happen. So we were thinking, what could be the signal that brings the pins to polarize? And from the work of other people and also us in the other parts of the plant, it's known that there is a positive feedback loop between the auxin and polarization of auxin flow, so meaning polarity of pins. So we just thought maybe it's really simple. You get just switching on the auxin biosynthesis at a given stage, meaning around the 16 cell stage. And then this will even more increase auxin levels, and this will polarize the pin and will trigger the whole process. So we started to look at the auxin biosynthesis in the embryos. And there are two main groups of the auxin biosynthetic or rate limiting factor in the auxin biosynthetic pathway. The one is the TAA family, which are three major members, and the others are the Yucca family. So we were looking at all of them, but basically the interesting thing was that at the 16 cell stage, we see all of the sudden in proembryo, here you see it by the GFP, it's the tagged version, so you see the expression of these auxin biosynthetic genes. You don't see it before. And after you see the expression of the auxin biosynthetic genes, you see the polarization of pin 1. So that was fitting quite nicely. And if you look in the mutants, you don't see the polarization. So, and this experiment is quite elaborated, actually, to have all the controls in, so I will try to explain you that. So this is a triple mutant for the auxin biosynthesis that is rescued by one of the members tagged with GFP. And it segregates. So you have embryos where you will have no GFP, and you know that they are defective in the biosynthesis because all three members are knocked out. And then in the embryos where you see the GFP, then you know that you have auxin biosynthesis because you rescued it by the construct. And so when Helen was calculating, she thought basically that there is 100% correlation. Whenever she gets GFP, she gets pin 1 polarization. In all the embryos where she don't get a GFP, she doesn't get pin 1 polarization. So it really looks like that the initiation of the auxin biosynthesis in the proembryo and this particular stage basically is triggering the polarization of pin and then doing the rest. So, of course, I realized that if you don't put a model, you don't get invited anymore to any meetings, so one has to do modeling. And so we just put our model on the auxin feedback regulation of pin polarity that I will not go into detail, which is here on the left. We put it on the embryo stuff. And then we didn't do anything else. So we didn't put any polarized pins. We just put a pin expression roughly everywhere. And that is the result. So basically, at the beginning, we assumed that all the embryo or the mother is producing auxin at the same rate. It didn't make a difference. And that made a polarization of pin 7 and then a weak auxin gradient in the proembryo. And then at the 16 cell stage, we simply switched on the biosynthesis here. Nothing else. All the polarity is done. The model makes the polarity like the apical polarity here in the protoderm, and then the basal polarity in the provascular cells. So it fits very well with the data. And at least within this model of the polarization in response to auxin, it seems that our assumption would be sufficient to generate both the correct polarity of the transport and the correct pattern of the auxin accumulation in the embryo. So now we are also trying to do the 3D templates and kind of move that all into the 3D, which in this case wouldn't be such a big problem because it's really axially symmetric at this stage. So there is no major issue with that. So just to summarize it. So we just have now data and model that would suggest that if you at a given stage of embryogenesis, they wanted to wake me up? They wanted to wake you up. So at a given moment of embryogenesis, you get biosynthesis, and we have the several UCA and TAA genes that are switched on at this stage. And if we don't have it there, we don't get the polarization. If we get it there by the feedback regulation, we get polarization of pin1, and that will make the polarity and maximum here. So it's really tempting to imagine that you would have also a biosynthesis in the mother, and basically mother at the early stage would polarize the embryo. And you wouldn't need anything else. You wouldn't need any additional patterning cues or spatial cues. Just the mother would produce embryo that would polarize pin 7 in the suspensor. And that would make the accumulation of auxin here, because there is nothing where it can go away, so it will always accumulate in the last cell of the file. And then that will undergo the division and differentiation, and within the program of the differentiation at given point you will have switched on the biosynthesis there, and that will swap it all other way and will synchronize, will signal across the clonal boundary and will synchronize the root meristem initiation with the proembryo development. So this is just what we have now. The model fits, the data look okay. It might be true, it might be not true. There are certainly additional factors, but that would be in theory sufficient. And so that was how endogenous signals, which we believe in this case auxin, change the polarity of pin localization and change then the auxin direction of auxin fluxes. Malcolm was speaking about the gravitropism as an example where the exogenous signal, so environmental signal, is translated into the growth response. Another classical signal is phototropism. So in both cases, you get directional signal, light coming from here, or gravity being, well, where it usually is. And then you get the asymmetric auxin distribution mediating directional growth and bending. And so in both cases, what we could see is that light in this case generate asymmetry in the pin 3 localization. That's the hypocotyl endodermis cells. In the dark, it's symmetric, both sides the same. When you put a light from one side, here it starts to disappear. Here it gets stabilized, and that would be enough probably to generate the asymmetry in the flow. So auxin coming from the top would be partially diverted to the shaded side where it accumulates, promotes growth, bending. And in case of gravitropismus, Malcolm was quite elaborate on the statolith sedimentation. So we know that the statolith sedimentation is happening, and together or later or before, we don't have it really temporarily resolved. We have a polarization of pin 3 that originally was, so this is a pin 3 in columella cells, originally is on all sides. Once you turn it pretty quickly, actually within 5 minutes already we can see the start of the asymmetry. So you get the asymmetry of the pin localization at the lower side, which would then divert the flow to the lower side of the root. Here, pin 2 would take over, bring it to the elongation zone, inhibiting elongation, again, bending. So these are the examples how the environmental signals via changes of pin localization can divert auxin fluxes. So just summarize it. So we have these, as I said, these cells that have the capacity to change the polarity of the pin proteins and therefore change how much auxin flows to which direction. There is a quite elaborated subcellular dynamics, so constitutive endocytosis in recycling that I didn't speak about, but this system can integrate these different signals and translate them into the asymmetric auxin distribution. So, as I mentioned at the beginning, we use also this model to study processes like endocytosis, recycling, sorting into the vacuole, and in particular polarity. So how these proteins generally in plants are getting to specifically one side of the cell, how they stay at the side of the cell, and how then the polarity can change in response to these different signals. So I will just now speak no auxin anymore, just simply the issue how the protein can stay within the polar domain and not go anywhere else. And this is not really a trivial issue, because in animal cells you have these specialized structures in the cell. So this is a Drosophila epithelium cell. In green you have an apical domain, and with red you have basolateral domain. These two domains are separated by the so-called tight junctions, which are forming diffusion barriers in the plasma membrane. So once you get a protein here or you get a protein here, they cannot cross the boundary and they stay within the correct polar domain. In most cell types in plants, we don't have tight junctions, maybe with exception of the endodermis cells and Casparian strip, but in all the rest, we didn't detect anything similar. So that's the one main difference between animal and plant cell concerning polarity. We don't have these diffusion barriers. The other thing is that in the plants, it seems that we have a more elaborated system for sorting polar proteins, because we could have in one single cell proteins at the lower side, upper side here in blue, and at the lateral side. So you have much more elaborated polar sorting going on that is known from animal cells. And so I will now try to tackle the problem how to keep the protein in place without the junction. So Jürgen, that's the guy who did it. He is now, since half a year, a group leader in Vienna and pursuing his own luck and fun. But just before he left, he did this great work, very nice microscopy, just looking how the pins are arriving to the plasma membrane, what happens to them once they get there, and how they are getting away. So this is how they are arriving. So he was able to visualize the, whatever you call it, recycling endosome trans-Golgi network. These are the structures, how they arrive to the plasma membrane. And he found out that they preferentially arrive to the middle of the polar domain. So if you look at the polar domain from the top, that's the highest intensity of the pins in red, and then yellow, green, and it's getting lower and lower. So if you look at the polar domain, the most pins are in the middle, and then you get a gradient in the membrane. You have less and less towards the border of the polar domain. So that's the first thing. So they arrive to the middle. The second thing, and this is related to John's work, John Runyon from Oxford Brookes. He was speaking a few talks ago, and he's also collaborating with us on this topic and helped us a lot. So this is about once the pins arrive to the plasma membrane, what is their mobility? Normally, you would expect within the very fluid membrane, the mobility of the protein would be high, and John showed you that this is not necessarily true, and there are different proteins having different mobilities. So definitely for pin, the mobility is very limited. So if you make the frap, so meaning you burn away the hole, and then you are looking how fast the hole gets closed. Can you dim it again, please? So it's better if most of you sleep, and the ones who don't sleep will see what I want to show, I think. So that's the hole, and that's the pin that wasn't burned, and it should close the hole. And if you look at the time, it's closing the hole extremely slowly, right? So there is a small fraction, that's the green stuff, which closes it immediately, as you would expect from the fluidity of the membrane. But the majority of the pin really walks in very slow. And so when we use the super resolution microscopy, we found out that the pins in the plasma membrane are localized in these clusters. We call them clusters. We don't know what they are. So that's how they look like. And they don't move. If you make the imaging over time, you see they are staying at the same place. So, and majority of the pins is recruited into those clusters. When we were comparing our pins, whether apical or basal localized, doesn't matter, to other plasma membrane protein, we couldn't see the similar cluster localization of proteins like aquaporin or the auxin influx carrier Malcolm-like so much. So I think that's something what is more specific for the pin proteins. We don't know what they are. They are a bit sterol dependent, because when we deplete sterols, the recruitment of pins into the clusters disappears, but that's all we know. So that was then what happens to the pins when they arrive to the plasma membrane. And then how they get away in the last part. So Jürgen looked at the edges of the polar domain, so here in these corners. And he found out places in these edges where he saw regular arrival of endosome and sequential arrival of endosome. So you have endosome coming, cleaving, another endosome coming, cleaving, and that always to the same place at the plasma membrane, right? If you look what happens to the pin at the place where the endosome arrives, so that's here. So when the endosome arrives, you see that there is a drop in signal intensity. So from red to, well, what is it, green. So that's the drop in the signal intensity. So at the moment when the endosome arrives, there is a lot of pin disappearing from the plasma membrane. So that is, of course, suggesting very strongly that the pin is getting sucked into the vesicle or into the endosome, and the endosome goes away. So it would be like a bus stop. Pins waiting at the bus stop, empty bus is coming and leaving full of pin, and the next bus coming, leaving full of pin, like that all the time. That would be the idea. We could also show how the pins are going into the buses by this approach. So if we paint by the laser the pin at the plasma membrane red, and we have the endosome marked green, at the moment when the endosome comes, it becomes yellow, indicating that the red stuff came into the green bus. So here basically what you really see is endosomes coming by probably burst of short-range vesicle traffic, getting the pins and leaving. And that happens preferentially at the edge of the polar domain. So one would assume that the pin, which despite all this recruitment to the clusters and limited lateral diffusion escapes from the polar domain, then gets internalized here and then recycled back to the middle of the polar domain. And like that you can keep the polarity. So altogether we have superpolar exocytosis, or rather recycling, because the original secretion of the de novo synthesized protein is apolar that we showed some years ago. So this is recycling, is polar to the middle of the polar domain. Then we have these clusters slowing the diffusion, and what escaped got internalized at the edges and recycled. And all this together then keeps the pins at the polar domain. And we again modeled that, so we divided the membrane to the clusters, and we put the ratios of the recruitments and all these things, and we put all these three processes. And we found out by the model that this is the most critical one. So these two alone will not will not maintain the polarity. So even with a slow diffusion and these, at some point you will get lot of lateral signals and the polarity will be not maintained. But if you have this endocytosis specifically at the edges, then the system works and you can maintain the polarity. Okay. Beside the microscopy, we do also forward genetics. And that's the picture of Elena who did that before the screen. She didn't want me to take a picture after the screen. And she was looking for mutants that would have defects in pin polarity. And because I really didn't want to lose her, she's extremely great in the lab for many reasons, which I'm not going into details. I didn't want her to screen all these seedlings under the confocal microscope looking for the polarity defect. So she did this clever screen that we generated some years ago, a line where the pin is localized on the opposite side of epidermis cells. And as a consequence, these seedlings are agravitropic. So we mutagenized this line and were hoping that if the polarity will be messed up, you get at least part of the pin at the correct side, upper side of the cell, and that will rescue the gravitropism at least to some extent. And so like that, we translated the polarity problem at a cellular level to the morphological problem, which you can screen by your eye looking for the mutant, which will be gravitropic again. And this is a typical plate, well, not really typical, because on most typically you don't have a positive one. So, but this is one with a positive hit. So this would be the seedling that shows the gravitropic response. Elena was turning twice, so therefore you have this kick here. This would be the positive control in the blue, and that would be the hit, which then Elena's husband, the basketball teacher from Romania, mapped. I mean, I don't know how he plays basketball, but he's mapping really, really well. He maps all our mutants. So he mapped this one. Of course, the mutant has the rescue of the gravitropism, it has rescue of the polarity, and it's a cell wall component, so it's a cellulose synthase 3, which was originally a big disappointment because we wanted some really polarity component. But then we found out that maybe cell wall and polarity, it's not such a stupid idea. And just very quickly, when we dissolve the cell wall, the polarity is gone immediately. And when we plasmolyze, then the polarity would be also gone immediately. And in addition, we found that the polar domain is connected to the cell wall by something, and that is very nicely coming together with what John showed for his formins and the connections between the cell wall and the plasma membrane. And so this was nice to see when the microscopy and genetics basically came together to reveal something about how plants can maintain polarity in absence of tight junctions. And I would like to thank the group. They are fine. I hope so. I like them. Well, I don't know whether they like me. And I thank you for your attention.